Assalamu alaikum. And third component of recombinant DNA technology is molecular carriers or vectors. Students, vectors are used to transfer our gene of interest into an expression system. Vectors, different kinds of vectors, they can be used in recombinant DNA technology. The one which is mostly used, that is circular DNA molecule of a bacterial cell, which is called as what, plasmid. Or some other kinds of vectors, they can also be used, like phage DNA. Phage DNA means like bacteriophages DNA. Bacteriophages are actually viruses which attack on that of the bacterial cell, so their phage DNA that can be used. Or sometimes cosmids, they are used. Now, what are cosmids? They are combination of plasmid and phage DNA, and sometimes yeast artificial chromosomes or yaks, they are also used for this purpose. Now, what are the characteristics of a vector? Of cloning vector, it must has origin of replication sites, means it is having the potential to replicate itself inside the expression system. Secondly, it must have antibiotic resistant genes on it, so it can be isolated when we insert our gene of interest into that, and we pour the vector into that of a medium containing antibiotics. So if it shows resistance against that particular kind of antibiotic, then it means this is our gene of this is that vector which is containing our gene of interest. Third, it must have restriction sites of different enzymes. It can show resistance against against different enzymes. Now, number four, a component which is required in recombinant DNA technology, that is molecular glue. Student, molecular glues are a kind of molecules which can join our gene of interest into that of our vector, and that's why we call them what, glues. And mostly molecular glues which are used in recombinant DNA technology, that is DNA ligase. DNA ligase is used to join two different DNA fragments, like our gene of interest and the plasmid DNA molecule, or plasmid DNA. And then number five, components of the recombinant DNA technology is expression system. A suitable organism that can act as host for the recombinant vector to express itself, and that system is called as what, expression system. Student, an expression system must be a system that is having short generation type, like a bacterial cell that can be used for this purpose, because a single bacterial cell, it can divide after every 20 minutes, and it must be having simplicity of its genetic system. Its genetic system must be simple. That's why usually we prefer a bacterial cell, or bacterial cell that is considered as an ideal expression system, because it is having short lifespan or short generation time, plus it is a very simple genetic system as well. That's why mostly our recombinant DNA, we introduce our recombinant DNA into that of the bacterial cell. Now, with the help of diagram, you can see this is bacterial cell, and this is its circular DNA molecule, which is called as what, plasmid. First, we will isolate this plasmid from that of the bacterial cell, and gene of interest, that will be isolated from the desired cell. For example, if this is a pancreatic cell, so we can get insulin gene from the pancreatic cell, and then we can cut this gene of interest with the help of what, molecular scissors, which are called as what, restriction endonucleases. We will cut the same we will use the same enzyme to cut open the plasmid, to make the same sticky ends, or to make compatible sticky ends and then with the help of molecular glue we can join both gene of interest and plasmid DNA molecule, and then we will introduce this recombinant DNA into that of the expression system, which is a bacterial cell. And when bacterial cell, it will divide into two, our gene of interest that will divide along with the division of what bacterial cell. And so either multiple copies of that particular gene they can be obtained, or we can extract our desired protein from that of the expression system. So this is all about what recombinant DNA technology. Now student, a very important point, how can we isolate or how can we identify our transformed clone. We can identify our transformed clone by adding a particular antibiotic for which resistant genes they are found on that of the plasmid into the medium. As the transformed clone has got resistance against the antibiotic, so it remains alive and continues to grow, whereas at all the untransformed clones they will be killed by the antibiotics. From this, scientists they can isolate their transformed clones from the medium. So this is all about what recombinant DNA technology. Thank you students.